Good day, everyone. Now we are on our final period, and the first lesson for your finals would be all about your renal function. So the actual PowerPoint was made by Professor Orig, and I will be using the same PowerPoint as he did, which is a little bit of edits lang pod gamay. So this will be our objectives, but I will no longer be reading it one by one. The objective stated here is summarized into our topic outline. So we will be discussing about your renal anatomy, then your renal physiology, then next would be your renal function test, and last would be about your non-protein nitrogen. Now, in this lesson, in this video lecture, kids, we'll divide this into two parts. Bale, for the non-protein nitrogen compound, so sa second part na lang po na siya kay medyo taas-taas po na siya ng discussion. So, for this video lecture, we will be focusing on the first three parts. Starting with your renal anatomy. Now, in terms of clinical chemistry, since we are in clinical chemistry, we will not be focusing more on what kidneys really all about. So we'll give way or we'll give the deeper discussion of that to your AUBF na subject puhon-puhon. So basically, we will just be talking about the general concepts of your kidneys. So, bale, in terms of the general characteristics of your kidneys, it is actually a paired bean-shaped organs, which is found in either side of our spinal column, and your kidneys is about the size of a fist, which measures around 10 to 12 centimeters. Now, in terms of the specific location of your kidneys in our body, it is between your T12 to L3. So, this is actually referring to your spinal column, T for your thoracic, and L for your lumbar. As seen here in this image, so ito po, 12th rib, then po pointing, then ayan po yung kidneys nyo, between your T12 to L3. So, looking at the macroscopic anatomy of your kidneys, so we will just be focusing on these three main, your renal cortex, your renal medulla, and we have your renal pelvis. So as you can see in terms of the placement of these different parts, your renal cortex is the outermost region, whereas your medulla is the innermost. Now, both your cortex and medulla make up the functional tissue of your kidney. Your renal cortex being at the outer region of your kidney, it will be the one containing your blood vessels, which is connected to your nephrons, whereas your renal medulla being the innermost part of your kidney, it contains now what we call your renal pyramid. So as you can see, diba, as pointed here, this is your renal medulla, and then your renal medulla contains or makes up about 8 to 12 renal pyramids. These pyramids that you are seeing here, your renal pyramids, and that these pyramids contain almost about 1 million of your nephrons. Now, since we are done with cortex and medulla, how about your pelvis? Now, for pelvis, it is the central region. Diba? As you can see here, ito po yung parang renal pelvis na portion as pointed. Now, your renal pelvis is the central region of your kidney, and it is located in your renal sinuses. When we say sinuses, para siyang yung cavity inside your kidney, and that this renal pelvis being a large cavity, siya ang somehow dira makolek. That is where your urine is being collected. And as you can see, diba, diretso na siya. Renal pelvis is padiretso sa inyuhang ureter. So that is your three basic macroscopic anatomy for kidneys. Now we proceed with your microscopic. Now, as mentioned, uh, each of our kidneys, not actually kidney, but each of your renal pyramid would contain about 1 million of your nephrons. And that your nephron, as you all know, it is the functional unit of your kidney. Now, if you try to look at the placement of your nephron, kids, Uh, some part of it is actually found in your cortex, and then most part of it kay naa sa medulla. Kumbaga murag it runs at both your cortex and medulla. And if you try to talk about which specific portion of your nephron is found or seen in cortex and ano yung mga parts na nasa medulla nyo, your glomerulus and your convoluted tubules are located in your cortex, whereas the rest, especially your collecting ducts, they are located in your medulla. So, kaning nephron ninyo, syempre napod ni siya iyahang different parts. Bale, we can summarize these different parts into five. You have your Bowman's capsule, or pwede nato na siya i-slash into glomerulus. Actually, glomerulus and Bowman's capsule are different kids, so we'll just discuss that further. So first, we have your glomerulus and your Bowman's capsule. Then the second one would be your proximal tubule. Then you have your loop of Henle. Then you have your distal tubule. And then lastly, we have your collecting duct. So as stated, glomerulus and Bowman's capsule, they are different uh, parts of, they are not, dili na sila kumbaga isa or the same. They are different parts of your kidneys in which the glomerulus is a cluster or tuft of your capillaries, whereas your Bowman capsule is a single layer of your epithelial cell, is single layer na siya of epithelial cells. So you can see the tuft of capillaries you see here is your glomerulus. Tapos na siya gamay na space, and then ang nagasurround ana that is your Bowman's capsule. Mao na siya ang difference, and then you will further be able to appreciate the difference or function of each of this as we go on our discussion later on. Now, kindly take note also that there are actually two types of nephrons in the body. So as you can see here, this two that is boxed in red, these are your two types of nephron. You have your cortical nephron and you also have your juxtamedullary nephron. Now, about 85% are cortical nephrons, and the rest of the 15% they are your juxtamedullary nephrons. So just by looking at this image, you will already have a rough idea as to its difference. And actually, it is also stated here in the kaning naka-box unsa man ang ilahang difference. So very first is that your cortical nephron has a very short nephron loop, as you can see here. Unlike sa juxtamedullary ninyo kids na it has a very long nephron loop. Next difference is in terms of the placement of your glomerulus, wherein upon looking at your cortex medulla junction, of course with cortical nephron it is farther away, daraman inyong glomerulus, whereas for your juxtamedullary nephron it is nearer to your juxtamedullary cortex juxtamedullary junction. And lastly, uh, in terms of the capillary that is present, here in your cortical nephron, the efferent arterioles here supplies your peritubular capillaries, whereas here in your juxtamedullary, the efferent arterioles supplies your vasa recta. So, bale, what is the difference between your vasa recta and your peritubular capillaries here? If you try to look at the function of this, peritubular capillaries are those capillaries that surrounds your proximal and your distal convoluted tubule, and its main function is more on reabsorbance. Whereas for your vasa recta is a meshwork of capillary system as seen here in this image, as pointed here, o dara ang vasa recta ninyo, and it envelopes or it surrounds your loop of Henle. And in terms of its function, more on ang function ni vasa recta is in terms of the concentration gradient of our kidney. Murag kumbaga, isa ni siya sa naga-concentrate or naga-dilute, murag naa sa iyaha Nasa iya ang power na either maging concentrated or diluted ang ato ang urine. So that is why, looking at the function of the surrounding capillaries of these nephron, murag yung ana na pud na, parang ganun na rin ang function ni cortical nephron and ni juxtamedullary. Kumbaga with cortical nephron, since siya ang major, then siya ang more on nag-regulate, na regulatory and excretory na function sa ato ang body. Whereas for juxtamedullary, as stated, since di ba vasa recta man ang nagasurround po dira sa inyuhang loop, then it's more on the concentration gradient, control of your concentration gradient, whether it will concentrate or dilute your urine. So going to the microscopic anatomy, actually na naganitay rough na idea as to its microscopic anatomy because we are already talking about your nephron. So this is the different parts of your nephron. So as stated, you have your first five, your glomerulus, your proximal convoluted tubule, your loop of Henle, your distal convoluted tubule, and then lastly, your collecting ducts. In terms of your glomerulus, before it will actually go to your glomerulus, natay duha ka different arteriole dire na murag dako pud og role in terms of the function, in terms of para mag-function well po jud or mabuhat sa inyong glomerulus ang iyahang function. So we have the very first uh the very first part that would actually pass through here na para inyuhang blood mag-pass through padulong gyud sa inyuhang nephrons, una sa gyud niya agian inyuhang afferent arteriole. Now, after afferent arteriole, it will pass through your glomerulus. That is what is being referred to as your tuft of capillaries, and then going to your afferent arteriole. Whatever is being filtered, kids, that will go to your Bowman's space and to your Bowman's capsule. Later, we will get to appreciate more of this. Napo ko i-present sa inyo in terms of unsa gyud ang whole mechanism aning ato ang nephrons. Then, syempre, once it passes through your Bowman's capsule, it will now proceed to your proximal convoluted tubule and then proceed to your loop of Henle. So there are actually two types of loop of Henle. You have your descending and ascending loop of Henle. And there are actually two, murag pwede pa nato ma-divide into two inyuhang descending and ascending. So you have your thick and thin descending loop of Henle as seen here. Na short lang ni siya. So, thick and thin din yung descending nyo. And then, you also have your thick and thin na ascending loop of Henle. Then, after passing through your loop of Henle, it will now go to your distal convoluted tubule, this one. And then, after your distal convoluted tubule, dun na sa inyong collecting ducts. So, as stated, to summarize, the function of your nephron, the function of each of those specific part of your nephrons, kindly watch this video, and if you think you already know enough, then you can actually just skip this part. Actually, grabe kagwapo ani na vid ang pagka-explain. It's very simple and baga simple lang pagka-explain pero taga na kayo gud mo makuha sa iya. So, kindly watch this video.
Hiya, welcome to Biomed Sessions with me, Ruse. Today we're going to be discussing the function of the nephron. Here we have our nephron, the unit of the kidney responsible mainly for the production of urine in order to get rid of excess water and toxic waste products. There are approximately 1 million nephrons in each kidney. At this end of the nephron, blood enters in via the afferent arteriole, passes through a network of capillaries called the glomerulus, and leaves via the efferent arteriole. As this occurs, components of the blood are filtered out into an area known as the Bowman's capsule. The filtered components, collectively known as glomerular filtrate, pass on into the tubules. Eventually, filtrate enters the last part, the collecting duct, and exits as urine. In men, the kidneys typically produce about 180 litres per day of this filtrate, yet only 1 to 2 litres of urine are produced each day. This means that only 1% of the filtrate manages to leave the nephrons. So what happens to the other 99%? For the most part, filtrate is reabsorbed back into the blood at several locations along the tubule. But how can reabsorption occur? Well, the efferent arteriole doesn't just end there. It branches out to form capillaries which surround the tubules of the nephron. Reabsorption is the movement of substances out of the tubules across the surrounding interstitial fluid into the blood of the capillaries. Now, whilst we're on the topic, we might as well discuss secretion, which occurs in small amounts in the nephron. This is the process whereby substances move from the blood through the interstitial fluid into the tubule. To make things simpler, let's remove the surrounding capillaries and zoom in on the first part of the journey where filtration occurs. I've covered this process in detail in a previous video, which I've linked for you to check out. But basically, most components of the blood, except for blood cells and the majority of proteins, are squeezed out between the pores of the glomerulus to reach the Bowman's capsule. So the glomerular filtrate contains substances such as water, glucose, amino acids and inorganic ions such as sodium, chloride, potassium and bicarbonate. It also contains the waste products creatinine and urea. Small amounts of these substances in the blood are okay, but they are harmful in large doses, hence the body rids itself of significant amounts via the kidneys. The next destination for the filtrate is the proximal convoluted tubule. Convoluted simply means twisted or coiled. This region is lined with simple cuboidal epithelium, and substances pass from the basolateral surface to the apical surface of the cells and out into the interstitial fluid. Approximately 65% of sodium ions are reabsorbed here through various methods. Positive attracts negative, so naturally wherever sodium ions go, chloride ions follow. Other negative ions do so too. As you may know, NaCl is the formula for common table salt, so it would be pretty salty outside the tubule if it wasn't for the fact that 65% of water leaves by osmosis. In addition, 65% of potassium is reabsorbed, as well as 90% of bicarbonate ions, which are important in maintaining the pH of the blood. But glucose and amino acids are the champions here, as around 100% are reabsorbed back into the blood to be used in processes such as respiration and making proteins. Note, not all urea leaves the nephron in the urine, as about 50% is reabsorbed. And secretion? Well, ammonia as well as various medications are secreted into the tubule. The next part of the nephron is the loop of Henle, which has a descending limb in which the filtrate travels down, and an ascending limb in which the filtrate travels up. The thin segments of the loop are lined with squamous epithelium. Now the loop of Henle dips down into a region of the kidney known as the medulla. This region is highly salty. Why? Because sodium is actively pumped out of the thick ascending limb, followed by chloride, but water itself cannot follow because the ascending limb is impermeable to water, hence the interstitial fluid becomes salty. The descending limb, however, is permeable to water, but has very low permeability to ions such as sodium and also urea. So as the filtrate travels down the descending limb, water leaves by osmosis because of the salty environment of the medulla. By the time the filtrate reaches the bottom of the loop, it is highly concentrated as significant amounts of water have left. However, the story is different in the ascending limb, whereby any water that remains cannot escape the filtrate. Sodium chloride, on the other hand, leaves passively from the thin ascending limb, then is actively pumped out of the thick ascending limb. After these processes have occurred, the filtrate is dilute. In total, approximately 25% of sodium chloride is reabsorbed from the loop of Henle. In order to avoid cell damage, the amount of water and salts in the blood needs to be kept regulated, and the nephron plays a vital role in this. The distal convoluted tubule, which is made up mostly of simple cuboidal cells, is the place where initial adjustments are made to the filtrate. Under the influence of the hormone aldosterone, sodium is reabsorbed, followed closely by chloride, whilst potassium or hydrogen ions are secreted into the tubule. Almost all remaining bicarbonate is reabsorbed, and although the distal tubule is normally impermeable to water, Small amounts may be reabsorbed here too. The collecting duct, which is composed of principal and intercalated cells, is where the final adjustments take place before the filtrate leaves as urine. Here, sodium chloride is reabsorbed in addition to urea, although sometimes urea can re-enter the tubule at the loop of Henle in a process known as urea recycling. But what about water? Levels of water in the collecting duct are regulated by the hormone ADH, which acts to retain water in the body by enabling it to be reabsorbed back into the blood. For example, if you are dehydrated, ADH allows aquaporins, aka water channels, to be inserted into the epithelium, allowing water to be reabsorbed back into the blood. Remember, the medulla is salty, so if water is given the opportunity to leave, it will. On the other hand, if you're overhydrated, a lack of ADH will mean that the collecting duct is more or less impermeable to water, so excess water can pass out into the urine. In general, urine contains water, urea, sodium, chloride, potassium and creatinine, as well as other inorganic substances. Okay, hopefully you found this tutorial helpful. If so, don't forget to like, and if you want to see more videos from me, do subscribe. Okay, see you in another video. Bye!
So as you can see, kids, actually, kani pa lang gani, this is just actually a 7-minute video and ang dami-dami niya nang na-cover. Diba? All the things that uh, this video discussed, mako-cover din natin siya. But then again, this is already an overview as to the role of each part of your nephrons. And then about the rest, about the terms, reabsorption, secretion, you will get to know more about that as we go along our lecture. Is, diba, mura na pug ka nang nagka-heads up na pud mo sa what's about to be discussed pa in this, in our lesson. So we are done with the anatomy. Now let's proceed to the second part of our discussion, which is all about your renal physiology. Now, when we say physiology, this is about, but this is the study of how our human body works. So basically, we will be talking about now the role of our kidneys, especially in relation to clinical chemistry. So starting with the function of our kidneys. So syempre, the first thing that would always come into our mind when we talk about kidneys is the urine. So that is why the very first function stated here is all about urine formation. So other functions of your kidneys would be in terms of balance, in which kidney maintains our fluid and electrolyte balance. Other than that, it also regulates our acid-base balance. Next, so you will get to know more about this acid-base balance later on when you reach sa inyuhang acid-base ata po jud na name sa inyuhang topic. And then next is all about, syempre, when we talk about kidneys, this is one way of excreting waste products, diba? So that is why in terms of role, na-adjud ning term na excretion. It excrete waste products, not just from protein metabolism. Actually, there are a lot of waste products that can be excreted, including your drugs and toxins. And then kidneys also have a big role in terms of secretion of hormones. So kidneys could also be considered as an endocrine organ since it has its own hormones na kanang ginasecrete, and these are the following hormones it is being secreted. So we have your renin, your erythropoietin, your 125 dihydroxyvitamin D3, and you have your prostaglandins. So just a rough description as to the role of each of these hormones. So when we say renin, kids, remember your RAAS, your renin-angiotensin-aldosterone system, in which from the name, di ba, parts sa renin dira. So basically, renin helps control our blood pressure and it also indirectly helps maintain the level of our sodium and potassium. How about your erythropoietin? So from the word, this word pa lang na erythro, So this is related to your erythrocytes or what we call your red blood cells. So basically, erythropoietin stimulate red blood cell production. What about your 125-dihydroxyvitamin D3? Now, this is actually just your vitamin D, but this is the complete name of the active vitamin D. Kaya naman pod ko'y inactive food na vitamin D na po'y inactive din. Siyempre si active ang inyuhang ang form na dapat na kailangan nato na para mag-perform gyud sa iyahang function. So for your vitamin D, this is involved in terms of your helping for absorption of calcium, more on sa calcium na role si vitamin D. And for your prostaglandin kids, prostaglandin is more on, mas ma-appreciate ninyo ni siya in terms of sa immune function. Kaya this is actually a pro-inflammatory, pro-inflammatory na substance. But if we try to relate it to kidneys, prostaglandins help in the regulation of our renal blood flow. So again, that is just a general description of the roles or function of these hormones being secreted by our kidneys. So let's proceed. So, other than the functions mentioned a while ago, we could actually summarize the basic renal process or the basic processes that our kidneys is being done by these four. Pwede nato siya i-summarize into four. So we have your renal blood flow, then your glomerular filtration, your tubular reabsorption, and then your tubular secretion. Looking at the different parts of your nephrons, murag kanina na iyahang pinaka-general na gyud na ginabuhat in order for them to function, in order for kidneys to maintain balance, to excrete unnecessary products, and for urine to be formed. So, let us start with renal blood flow. Siyempre, di ba, kidneys man gyud ni ang kanang kumbaga isa sa naga-clean sa ato ang blood. That is why kidneys is very important kay isa man ni siya sa kanang again mag-remove sa mga unnecessary substances present in our blood. And in terms of renal blood flow kids, around 1,200 to 1,500 ml per minute na ang ato ang renal blood flow. Now, if we start from the very top, siyempre mag-start mo yun sa inyong heart, diba? So, mo ni siya ang kumbaga murag kani path, renal blood flow path sa ato ang vessels. So, starting with your aorta, passing to your renal artery, your segmental artery, to your interlobal artery, then your arcuate, then your cortical radiate artery, then saka pa katong inyuhang afferent arteriole. Then from afferent arteriole, it will pass through now to your glomerulus. And then anything na dili ma-filter sa inyong glomerulus, exit padulong sa inyong efferent. And then from your efferent arteriole, it will either pass through your peritubular capillaries or your vasa recta, depending on what specific type of nephron we are talking about. Diba? Remember your cortical and your juxtamedullary. Then after that, it will pass through your cortical radiate vein, your arcuate vein, your interlobal vein, your renal vein, and then back to the heart through passing through your inferior vena cava. So, ingana na ang murag specific blood vessels that our blood will pass through in order to be properly being cleansed. Pagka naman po ang gwapo na term, cleansed by our kidneys. So now we proceed with your glomerular filtration. Now, of course, from the name itself, this is the role of your glomerulus in which it filters our blood and the product and kung unsa man ang iyang mo produce, mo na siya atong ginatawag na glomerular na filtrate. So, there are a lot of factors. So, there are a lot of factors that make our glomerulus the best site for filtration. So, we tend to ask, what are these factors? Diba? So, first factor is that there is a high pressure in your glomerulus. Now, this high pressure in your glomerulus, kids, is brought upon by the position of your glomerulus. Now, remember, it is a tuft of capillaries, which is a result of the position between your two arterioles. So, because of the presence also of these two arterioles, ay high pressure naman create. So, isa na siya ka-factor as to why it can filter our blood. Next would be its semi-permeability. Diba when we say permeable, meaning to say dali kayo mag-pass through ang mga different na substances. Now, glomerulus is semi-permeable, meaning to say it does not completely, it does not let all of substances pass through. Ang cause ng semi-permeability niya is that one factor, isa ka factor niya is in terms of the size. So, bale, the cutoff value of the molecular size that is able to pass through your glomerulus is up to 66,000 Daltons. So if the size of your substance is more than that, then it is not able to be filtered. Dili siya able na ma-filter and dili siya able to pass through your glomerulus. So again, it is semi-permeable. It can pass through everything except those that are large in size. Now, another factor is that the basement membrane surrounding your glomerulus is actually negatively charged. And since it is negatively charged, thus it will repel all substances that carry negative charges. Siyempre, dali lang makapass through ang katong mga substances that carries a positive charge. And then, take note that that your glomerular filtrate is around 130 to 150 ml per minute. And in order to measure our glomerular filtrate, natin ginatawag na glomerular filtration rate. Ano na siya? This is one way of checking or testing if our glomerulus is properly working, or not just our glomerulus but our kidneys in general. So, bale, with glomerular filtration rate, this is the volume of blood filtered per minute, meaning to say, glomerular filtration rate, it estimates how much blood passes through our glomerulus each minute. Ingana na sa inyuhang glomerular filtration. So, since we are talking about filter or filtration being the property or the basic function of your glomerulus, now, the next question, other than knowing factors of how it can filter, kung saan iyahang substances na ma-filter, so our next question would be what can actually pass through your glomerulus. So, those that can pass through your glomerulus are the following. So, we have your water, your electrolytes, your glucose, your amino acids, your urea, and your creatinine. Those that cannot pass through your glomerulus are the following. So, you have your plasma proteins, your cellular elements, and your protein-bound molecules. So examples of your protein-bound molecules are your lipids. Mga protein-bound biya na inyuhang lipids. And if you try to compare this following with this that can pass through, diba isa ka factor yun nila is in terms of size. Proteins are actually large molecules, and that is why those that are protein bound or the protein in itself cannot really pass through your glomerulus. So now let's proceed with tubular reabsorption. Now, tubular reabsorption happens about 90% in your proximal convoluted tubule. So that is why murag major tubule yun ninyo inyong proximal, since that is where most of the reabsorption process or function of your kidney happens. And when we say reabsorption, di ba, we are trying to take in. Kumbaga, after filtering from your glomerulus, after na ma-filter, there are substances being filtered na ginabalik siya sa atong body. And syempre, daghan taong pwede i-consider ana, but it might be that because it is still needed. It's just that ma-filter siya kay tungod gagmay og size, and yet kailangan pa din siya sa body. That is why kailangan na naay mo reabsorb balik sa iyaha and ibalik sa ato ang body. Mo man na siya ang tubular reabsorption. It happens when the substances from your tubules are moved back to via either your peritubular capillaries or your vasa recta and going back to our blood vessels. And balik na po na siya circulate sa ato ang body. So, you can see here, this is the summary of the amount of your different substances in terms of how much is being reabsorbed. So 75% are being reabsorbed of your sodium chloride and water, 100% of your glucose, almost all of your amino acids, vitamins, and proteins, variable amounts of your urea, uric acid, and ions like your calcium, magnesium, potassium, and bicarbonate, and then 98 to 100% of your uric acid, only to be secreted at the DCT or at your distal convoluted tubule na part. So about this secretion na portions, we will be discussing that later. So there are only about 1% of your glomerular filtrate that actually leaves our body because the rest is being reabsorbed while it passes along your different tubules and different ducts. Di ba remember ka to sa inyo ang video na na-explain na daghan kayo og mga gina-reabsorb and there are specific substances being reabsorbed sa specific portions po sa inyuhang nephrons. It's because kana po mga nephrons nato as explained also sa katong video ninyo kaganina, di ba na ay mga certain properties po. That is why kanila ang pwede makapass or kanila po ang kanang ilahang pwede i-reabsorb. So in terms of how tubular reabsorption occurs in your tubules, so daghan ta og factors ana. So be familiar with the terms osmosis, diffusion, and then kana inyuhang passive and active transport. Manina sila ng mga factors that could actually help in terms of the reabsorption or actually movement in general sa inyong mga different substances, whether they need to go in or out sa ato ang mga kaning part sa inyong tubules. So, what we need to consider also in terms of reabsorption, tubular reabsorption, is this is what we call your renal threshold. Now, when we say renal threshold, this is defined as the concentration of your substance in the blood above which the kidneys begin to remove it into the urine. So uh, we could actually compare renal threshold as mura siyang kanang limit sa ato ang urine. Na kumbaga, if there is an excess amount of this substance in our body, that's the time na mag-start na siya og appear sa ato ang urine. So let us take for example glucose. Renal threshold of your glucose is about 160 to 180 milligrams per deciliter. So above or beyond, or if it's greater than that, if ang glucose concentration sa atong body is greater than that, then that's the time that it will start to appear in our urine. Okay? Again, any excess amount in our substance, itry mag yun nato na siya og excrete. And one way to help in lowering this level of the level of these substances is kani po yung igawas nato siya through our urine. So that is why that's the time that they are na ma-detect na nato na siya could be through the use of your reagent na kaning urine dipsticks nato na mag-start na mo og positive ani na mga certain parameters. So remember, if a substance concentration exceeds the renal threshold for tubular reabsorption, it will now appear in the urine. So this is a summary. This table shows the summary of what specific mechanism it undergoes, what substances, and specific location of your nephron as to these substances are being reabsorbed. And actually, pwede na ninyo po makita na summary diri kaning sa inyo makita na image diri sa inyong right side na naay mga specific substances indicated sa taga-part sa inyong nephron in which siyempre pinakadaghan jud kay since major man gyud ninyo na tubule na naay reabsorption na naga-reabsorb sa inyong PCT. So mo ni siya ang mga naka-acronyms lang na kids. This is referring to your glucose, amino acids, your water, and then your urea. Siyempre water, then kani chloride, urea, and salt. And then still kani sodium rather. This is sodium. Sodium. Then additional reabsorption of water pa diri sa inyuhang collecting ducts. Then again, recall what is active and passive transport. When we say active transport, di ba, it requires energy because in terms of the movement, it is from a lower to a higher area. Whereas for passive transport, it does not need energy since the movement is from a high to a lower area. So, para ma-remember ninyo na siya, just imagine you walking up and down the stairs. But whenever you go from a lower to a higher na level, diba, mas kapoy siya. So, it requires more energy that is active. Whereas for passive, from a high to low na area, and di ba mas dali biya gyud pababa the stairs, and usually dili kay siya ingana ka effort, it requires less to no energy at all. Mao na siya ang murag kanang pwede ninyong isipon na concept to really remember what is active and passive transport. So, as you can see here, all of this stated here from the substances being reabsorbed as to why kana lang na portion ang gina-reabsorb, that is explained well in the video that I presented a while ago. Nakalimutan man, then just go back to that video kay para dili na lang pud confusing, so dito na to tanan. So kani another representation lang pod in terms of what is being reabsorbed in each of the part of your nephron. So this is just another representation lang pod. So we'll place that one in your handout, then just go over it and read it. So let's proceed with the next. Now, what will happen to your sodium and water balance? You could recall in the video, diba, there are already hormones being mentioned in the video in which there are specific areas also in your nephrons in which is being controlled by your uh different hormones. So remember your aldosterone and your antidiuretic hormone. So here aldosterone, which is basically sa distal convoluted tubule ninyo ang yang more on ang control in terms of reabsorption of your sodium. Then you have your antidiuretic hormone, which controls the level of your water, and that is ang affected ani na nephron or ang ihang gina-control is sa inyuhang collecting ducts or sa inyuhang collecting tubules. Now, the diseases stated here on the left side are actually related to the sodium and water balance that we are talking about. When we say Conn syndrome, just a general concept lang po, Conn syndrome is actually related to your aldosterone, in which when a person has a Conn syndrome, they are making too much aldosterone. And the main function of your aldosterone is in terms of controlling your sodium level. So that is why if there is too much aldosterone, there is also an increase in your sodium level. How about your Addison's disease? Now, for Addison's disease, they do not it's more on not producing enough aldosterone. So it is the opposite of your cons. Cons increase the aldosterone. For Addison's na decrease na aldosterone diri. How about SIADH and diabetes insipidus? This is related to your antidiuretic hormone. For SIADH, there is increased antidiuretic hormone. For diabetes insipidus, there is decreased antidiuretic hormone. And then again, the role of your antidiuretic hormone is that it controls your water. O more on water na siya na. If there is increased antidiuretic hormone, there is more water being reabsorbed. While for diabetes insipidus, since there is decreased antidiuretic hormone, then there is decreased water being reabsorbed. Yes. So, yun lang siya. So, ah, grabe ang role ni kidneys in terms of controlling these substances that we have mentioned. So now we proceed to the last part of our basic renal process, which is your tubular secretion. Now, tubular secretion has two definitions. One is that it involves the movement of your substances from your peritubular capillary to your tubules or to your tubular lumens. Kumbaga, tubular secretion is actually the opposite of your reabsorption. Eh kung si reabsorption from your tubules to your peritubular capillary, secretion will be the other way around. It is from your peritubular capillary to your tubules. Now, tubular cells secrete products of their own cellular metabolism to the filtrate in the tubular lumen. So there are actually two major functions of your secretion, your tubular secretion. One is eliminating waste products not filtered by the glomerulus. Diba remember what I have mentioned that the glomerulus via limited ang mga filter in terms of mga small substances lang. Now, what if there are large substances that cannot pass through your glomerulus but needs to be excreted out into our body? Then that's the time that your tubular secretion now will take up that role. So, kumbaga, siya ang magkuha ato ng mga product na wala na filter ni glomerulus na kailangan i-eliminate out sa ato ang body. So, other than that, tubular secretion also has this function in terms of regulating your acid-base balance in the body through the secretion of hydrogen ions. So, syempre, hydrogen ions are easily reabsorbed, and if there is a need to kaning in terms of the acid-base balance, kumbaga, kunwari na sobra-sobra na hydrogen ions, pwede ra pud mutabang og excrete out paana mga sobra na hydrogen ions ang kaning ato ang tubular secretion na function sa ato ang kidney. So, to summarize your filtration, deabsorption, and secretion kits, i-remember lang ninyo siya. Siyempre, for filtration, that is starting from your glomerulus until ma-secrete out na kanang normal passing through your different parts of the nephrons until na ma-excrete out na siya. With reabsorption, passing through your glomerulus, pero tubules back to peritubular capillaries and then back to our body. But for secretion kids, it is coming from outside. Tama ba ding image? It is coming from your peritubular capillaries going into your tubules and then being excreted out. Sorry, mali di ako ang gina-point out. For kaning substance A, this is referring to secretion. For substance B, actually almost related lamang po dyan ni si filtration o reabsorption, and as you can see in the image for substance B, this is your filtration. Okay, as you can see na ay arrow diri na going for excretion. O man gyud na ang ending sa atong gi-filter. Being filtered to be excreted. So for substance C here, this is actually referring to reabsorption. Since you can see, walay arrow going out into the urine. Baga, ang iyaha is going back into our body. Sorry ha, kaya wala na ako napansin na abi na akong naka-ABC, then referring na dayon to filtration, reabsorption, and secretion. So again, please do not be confused. Again, substance A is referring to the secretion process. Wala siya ni agi sa inyong glomerulus, nasa sa inyong peritubular capillaries, pero gibalik sa inyong tubules and then gi-excrete. For substance B, this is referring to filtration because substance is being filtered and then until it is excreted. For substance C, this is reabsorption since it is filtered in your glomerulus but then gibalik sa inyong peritubular capillaries until exit out balik sa atong body. So again, this summarizes your renal processes about filtration, reabsorption, and secretion. So now we go to the last part of this video, which is your renal function test. You might wonder na ma'am, 'di ba lecture man ta, why is it na naay mga test i-mention na diri? Now, actually this is very important for you to able to further understand this renal processes, your basic renal processes. And if you try to look at the test being mentioned here, 'di ba naka-refer gihapon siya sa atong basic renal na process. Muna na each of those renal processes napud tay ginatan-aw dira na mga test. Natay test na pwede buhaton in order for us to know if these basic renal processes are actually working. Muna natay glomerular filtration test, natay tubular reabsorption test, natay tubular secretion test, and we have your renal blood flow test. So, why do we need to perform this test? Syempre, isa man gyud the pinaka-main na reason ani is for us to really know if our kidneys are still functioning well. So this rely on the measurement of the waste products in the blood, which accumulate when kidneys begin to fail. Siyempre, this test rely on the measurement of waste products that accumulates when your kidneys begin to fail. So, siyempre, renal function tests are good tests since it would tell us that there is something wrong in our body. Since, diba, nabi atay mga substances that are not normally present in the urine. So, once gani na ma-detect na siya sa atong urine, then that is already very alarming. Meaning to say, nanay problem sa atong body, di ba kadugayan biya ana sa sige-sige ug try ug tabang sa atong kidneys na mag-balance o mag-normalize ang kung unsay naa sa inside sa atong body, magba pud kadugayan atong kidneys na ana. And that there should be 20 to 30% of the nephrons still functioning before the concentration of these products begin to accumulate in the blood. Diba? So that is how important knowing if our kidneys are also functioning well. Again, agi ba agyud dira ang atuang blood, diba mo man na natay renal blood flow, gina-cleanse dira sa atong kidney. So it is one way of knowing if there is really also a problem inside our body. So let us start with your glomerular filtration test. Now, test being performed here in order to check if our glomerulus is functioning well is what we call your clearance test. So this is a standard test used to measure the filtering capacity of your glomeruli. It measures the rate at which your kidneys are able to remove a filterable substance from the blood. So from the name itself, clearance, Ona, diba, it is related to saying the removal, what is being cleared off. So what we are looking at here is how well our kidneys are able to remove such substances from the blood. And there are actually a lot of tests to check the filtering capacity of our glomerulus. Though kanina portion kids, kanina portion is not really that common, but then it was mentioned in the book. It's nice to know na kasi magugma question mo na basig kanina mga substances are they being used to test glomerular filtration, then wala na noon mo kabalo because it is not uh, commonly used nowadays. Still, it is good to know. So, bale na amoy daghan, but then I summarized this into 8. You have your urea and creatinine clearance. These two will be further discussed in your laboratory, and when we reach your non-protein nitrogens na discussion, we have your inulin clearance, your cystatin C, which is a more common na test, and then you have your beta-2 microglobulin, your beta-trace protein, your tryptophan glycoconjugate, and then you have your NGAL. Now, before I would get to discuss at least general na description lang po about each of these tests, you need to know first uh, what substance should be analyzed for us to be able to test properly the filtering capacity of your glomerulus. Stated here, to ensure accuracy of the test, substance to be analyzed must be of the following. So this is actually our criteria. First, your substance should neither be reabsorbed nor secreted. Siyempre, ang ato biyang kinatanaw di ba is ang filtering capacity. So how would we know if the glomerulus filter substances properly if it is being reabsorbed or secreted in our our body rather. So if it is being reabsorbed or if it is being secreted, then we are looking at a different function na. So that is why we need to have a substance that is neither reabsorbed nor secreted. Agad-agad lang siya ma-excrete into our urine. Next criteria would be it should be stable in your urine during a possible 24-hour urine collection. Next, it should have a consistent plasma level. It should be available to the body as much as possible. It is already present in our body so as to prevent any harm or toxic related na kanang mga effects sa ato ang body. So as much as possible, it is already a part of our body. And last criteria would be should be available for chemical analysis or it can be tested. Or kumbaga, there should be already an available test that can be done for us to be able to measure the substance. Kasi it defeats the purpose na, sige, nalagi siya aning upat ka criteria, and yet pag-abot diri, dili day nato siya ma-measure, kay wala day tay any means to measure the substance. So, mahimong useless. So, as much as possible, there should be an available test to measure that substance. So, going back to our different clearance test here. Again, I won't be discussing all of it. We'll just point out mga important lang na mga facts about sa ilaha. Again, for urea and creatinine clearance test ninyo, we'll further discuss that one either sa inyuhang lab or at pag-abot sa atong non-protein nitrogen na discussion. Not sure pa manggod kung ato ba siyang ma-cover. So, dirita kay inulin. Inulin is actually regarded as your gold standard. The problem lang with inulin is that it requires an intravenous infusion, meaning to say, -1 na siya sa atong criteria na in terms of available siya daan sa atong body. Because again, it is being given intravenously. Nindot si inulin kay dili siya ma-reabsorb, dili pud siya ma-secrete. But the other disadvantage, other than katong gimension na it is not available in the body, other disadvantages of inulin is that it is expensive and the procedure or the test that is being performed to measure inulin is very time consuming. That is why di siya common yun na ginagamit na. Again, wala naman dyan lain. This is actually considered as your gold standard. Now, next would be your cystatin C. Okay. Cystatin C is available in the body because this is an inhibitor of your cystine proteinase, available in the body in such a way that it is being produced by all of our nucleated cells. And other advantages pa sa inyong cystatin C that it is not affected by age, by sex, or even by muscle mass, and it is neither reabsorbed nor secreted. It is readily filtered by the glomerulus because it has a very small size and it is positively charged. Now, the problem with cystatin C is that it is very difficult to measure your cystatin C and it is actually also expensive like your inulin. And isa actually sa nabasa na ako, kids, is that cystatin C, mali dito kung gi-mention na neither reabsorbed nor secreted. Sorry. Let me correct that one. The problem or disadvantage of your cystatin C is actually it is being reabsorbed. Now, why is it being tested? It's because once cystatin C is present in the urine, it would already give us an early na kanang na mura siya naga-signify og na murag early identification of a possible na kidney failure. Again, take note, cystatin C, let me just correct na it is actually completely reabsorbed. It's just that it is being tested because once cystatin C is present in the urine, it would already give us or it would already signify an early probable kidney failure. Then again, naman ko yung mga daghan pa nagipang mention pud sa inyuha na disadvantages that it is difficult to measure and measurement is also very expensive. Now, kaning mga beta-2 microglobulin, beta-trace protein, and kaning tryptophan glycoconjugate, actually mao-maura ang mga description ani. So, dili na lang ta kay NGAL. Kaya actually, naay gwapo pud na feature inyuhang NGAL. NGAL stands for, this is the meaning of your NGAL. This is your neutrophil gelatinase associated lipocalin. That is how it is pronounced. Neutrophil gelatinase associated lipocalin. Now, your NGAL is a protein being expressed by your neutrophils, including those epithelial cells present in your proximal convoluted tubule. Now, the good thing about NGAL is that, kumbaga, if there is an acute kidney injury, an acute kidney injury, it immediately rises. Baga, ang concentration ni NGAL, mag-elevate na yun siya within 2 to 6 hours. That is why it is a very useful, it is a very useful predictor. It is a very useful early predictor of your acute kidney injury. Since again, paspas kayo mag-increase ang iyang concentration. Mo nang isa ka advantage sa inyuhang NGAL. Then again, sa disadvantage lang po sa inyuhang NGAL, hindi siya ana ka-specific because in terms of specificity, NGAL manggod could also rise from other na mga diseases such as kung naay mga systemic stress, mga ingana. In the absence systemic stress in the absence of your AKI, it would be best to emphasize that one. In the absence of AKI and then namoy other na mga sakit, pwede mag-increase po din yung NGAL. Sige, so now we proceed to the next. Now we go to your tubular reabsorption test. Now, tubular reabsorption is the first function to be affected if your patient has renal diseases. If the measurement of your GFR is a useful na kanang indication na naay early na mga renal diseases, ang kaning sa law sa inyuhang tubular reabsorption capability, kana gyud pod ang first function affected sa naay mga renal disease. And the test here in order to check for tubular reabsorption is what we call now your concentration test. Clearance test kaganina sa inyuhang glomerular filtration. For tubular reabsorption, that is what we call your concentration test. Now, this concentration test determines the ability of your tubules to reabsorb the essential salts and water that have been non-selectively filtered by our glomerulus. And these are the following tests that can be done in order to check for your tubular reabsorption. So, kumbaga, we could actually relate the term concentration in terms of the ability of our tubules to either concentrate or dilute our urine. Kasi syempre, di ba, as stated in the video also a while ago na ang makapag-concentrate or dilute biagud sa atong urine is in terms of the number of substances present, including the number of water that's being excreted. So, muna concentration test ni siya. So, starting with osmolality and osmolarity. Now, syempre with osmolality and osmolarity, this measures the concentration of analytes in the urine. The difference lang between osmolality and osmolarity, when we say osmolality, it is the number of osmoles of solute in kilograms, whereas for osmolarity, we are looking at the number of osmoles of solute in a liter. Basically, it differs in terms of the unit. Kani osmoles per kilogram for osmolarity, this is osmoles per liter. How about your free water clearance? Actually, ang name niya na anadara sa name, ang kung unsa ang test about, this measures the amount of solute-free water being excreted in the kidney. And then for your Fishberg test and Mosenthal, this is more on in relation to checking the specific gravity. Again, for Fishberg and Mosenthal, this is looking at the specific gravity, in which for Fishberg test kids, natay 24-hour water deprivation na ginabuhat ani. So patient will be deprived of fluid or water for 24 hours, and then after which we will measure the specific gravity of the urine. Whereas for Mosenthal, this is day versus night, daytime versus nighttime na checking of your specific gravity. So it is comparing the volume and specific gravity of your urine sample in the morning and in the evening to evaluate the concentrating ability of your kidney or your tubules. So, mo na siya ang inyuhang concentration test. So now we proceed with your tubular secretion and your renal blood flow test. Now, to measure the exact amount of blood flowing through your kidneys, it is necessary to use a substance that is completely removed from the blood rather than being removed when the blood reaches the glomerulus. Total renal blood flow through your nephron must be measured by a substance that is secreted. Again, balikon lang po na ako, that the substance should be being secreted rather than being filtered by your glomerulus. Now, the principle of this test is actually almost the same with your clearance test katong sa inyong glomerular filtration. But however, to ensure measurement of your blood flow through the entire nephrons ninyo kids, substance as stated must be removed from your blood capillary rather than being removed when it is being reached in the glomerulus. Mo na somehow related inyong renal blood flow o tubular secretion test. And these are the specific tests being done to measure both. So you have your PAH test or your para-aminohippuric test. You have your titratable acidity and you have your urinary ammonia. Now, para-aminohippuric acid test is actually a non-toxic substance. Kumbaga, ginatake nato ni siya. Sumona ang isa ka disadvantage niya kay exogenous man gud siya na substance. It is not readily present in the body. It is being taken up. So, kaya siya exogenous. Walay problema, it is a non-toxic substance and it is actually loosely bound to plasma proteins. Now, with amora man to, actually for para-aminohippuric test, take in nato na siya and then check lang po nato atong urine. Pila ang amount of para-aminohippuric test ang naagihapon, ang na-retain. Working nga na ang concept ana na test. How about your titratable acidity and your urinary ammonia? Now, when we say titratable acidity, it measures the amount of acid present in a solution. Murag ang titratable lang ana na term kids is in terms of the process done in order to measure acidity. Murag titratable acidity. Now, take note that the ability of your kidney to produce an acidic urine rather, to produce an acidic urine greatly depends on the tubular secretion of your hydrogen ions and the production and secretion of your ammonia by the cells in your distal convoluted tubule. So as a result, Diba, small in molecular size via ang hydrogen ions ninyo and even your ammonia, they are being readily filtered and reabsorbed, and actual excretion of excess hydrogen ions depends on your tubular secretion. I guess murag na discuss man nina ko kaganino or dito ba sa video na, di ba? Acid-base balance ato ang ginatanaw ani if there is a need to excrete more hydrogen ions, then magdepende na siya sa inyong tubular secretion. Now, kaning titratable acidity kids and kaning urinary ammonia, this test can be run simultaneously. Kumbaga, kuha mo og naamoy ipainom ana sa inyong patient, your patient will be given an acid load na nag-consist og ammonium chloride pod, and then mag-collect mo ana og 2-hour interval na specimen sample. So, bale, before ug after ni siya, so wala pang gipainom atong oral na ammonium chloride. Then after 2 hours, uh, ah, kum- wait, collect sa mong ihi, after which, painomon ninyo siya, atong oral na ammonium chloride. And then after 2 hours, collect na pod mong ihi. So by titrating the amount of free hydrogen ions, and then the total acidity of the specimen, ammonium concentration can now be calculated. So, bali duha. Titratable acidity, i-measure sa ninyo. And then, after measuring the titratable acidity, i-measure na dayon ninyo ang total acidity. So, the difference now between these two would already be our ammonium na concentration. Muna siyag mahulog na ingani, kids. Muna, di ba, simultaneous ang duha. Your urinary ammonia would be equivalent to the total acidity minus the titratable acidity. Titratable, which is referring to the hydrogen ions here na present. That is how we would be able to get the concentration of your ammonia. Murag indirectly getting the concentration. Ano siya ang concept sa inyong tubular secretion and renal blood flow test. So that actually ends the first part of our lecture here. Next part would be all about your non-protein nitrogen. Break sa tagamay, and then after which, watch na dayon ta sa atong part 2. Thank you for listening, kids.